In 1977, the Japanese government approved a compound extracted from a mushroom for cancer treatment. It was prescribed alongside chemotherapy in hospitals across Japan. It improved 5-year survival rates in gastric cancer patients from 60 to 73%. By 1987, it accounted for 25% of everything Japan spent fighting cancer. Annual sales reached $357 million. The compound is called PSK. The mushroom it comes from grows on dead logs in every forest in the United States. A 60-day supply of the extract costs less than $30. The FDA has never approved it. Not because the science failed. The science was published in the Lancet. But because you cannot patent a mushroom, and no company will spend $1 billion on a drug approval for something it cannot own. A quiet moment in a Japanese neighborhood sparked the journey that would change cancer care for thousands. In the mid-1960s, a chemical engineer at Kureha Chemical Industry Company noticed his neighbor preparing tea from a fan-shaped mushroom growing on a decaying log. The neighbor, recovering from a serious illness, credited the homemade infusion for his resilience. Curious, the engineer collected a sample and brought it to the Kureha laboratory. There, the mushroom was identified as Trametes versicolor, a species known for its colorful layered caps and its ability to thrive on fallen wood across forests in Japan and beyond. Inside the lab, researchers began cultivating the wild specimen under controlled conditions. They assigned it a simple label, CM101. This unassuming code, entered into a field notebook and later preserved in a university culture bank, would become the foundation for the new line of investigation. Over the next several years, the team refined their extraction process, using hot water and ethanol to isolate a complex protein-bound polysaccharide from the mycelial culture. In 1971, the Kureha Research Group documented their findings in an internal technical note, officially naming the extract polysaccharide K, called PSK, to set it apart from other mushroom-derived compounds under study at the time. The original CM101 strain and its field record remain archived, their pages marking the transition from folk remedy to pharmaceutical research. What began as a neighbor's quiet ritual became a carefully cataloged scientific pursuit, setting the stage for what would soon become one of Japan's most prescribed cancer therapies. Long before laboratory notebooks and clinical trial protocols, the mushroom known as Trametes versicolor held a quiet place in the healing traditions of Asia. In the earliest Chinese materia medica, the Shenong Bencao Jing, a fungus called yunji, which means cloud mushroom, was described as a tonic for vitality and longevity. Later, Li Shizhen's Compendium of Materia Medica, compiled in the Ming Dynasty, listed yunji among the agents said to clear heat and to detoxify, though these texts made no mention of cancer. In Japan, the same mushroom appears as kawaratake, called mushroom by the riverbank. Edo period herbalists praised kawaratake for supporting recovery after illness, often preparing it as a simmered decoction for those weakened by fever or surgery. These records reveal a pattern. The mushroom was valued not as a cure for disease, but as a gentle restorer, a means to strengthen the body when it was most vulnerable. Beyond Asia, indigenous peoples in North America recognized the utility of turkey tail and related polypores. Cherokee and Ojibwe healers brewed mushroom teas for coughs, stomach trouble, and as a cleanser after ceremonial fasting. Ethnobotanical surveys from the 20th century document these practices, yet none describe systematic use against tumors. Instead, the mushroom's role was to support resilience, to help the sick regain balance after hardship. These centuries-old traditions did not predict the specific immune effects later uncovered in the lab, but they did establish a lineage of trust. The mushroom's reputation as a tonic, a protector, and a gentle companion in recovery would quietly echo into the scientific era, laying a foundation for the rigorous study that followed. By 1973, the Kureha team had moved beyond the bench, filing a Japanese patent for their extraction process. The method they protected was no simple kitchen recipe. It began with large-scale fermentation of the CM101 strain, grown in stainless steel tanks under tightly controlled conditions. Once the mycelium reached peak density, technicians transferred the culture to massive hot water extractors, where heat and time coaxed out the complex polysaccharides. Ethanol precipitation followed, separating the protein-bound fraction that would become PSK from less active components. Quality was not left to chance. Each batch passed through a three-step cascade. First, genetic fingerprinting confirmed the identity of the CM101 mycelium. Second, high-performance chromatography measured the purity and molecular weight of the polysaccharide fraction, ensuring over 90% beta-glucan content. And finally, each lot underwent rigorous endotoxin testing, with limits set below 0.05 EU per milliliter, stricter than many contemporary antibiotics. These checkpoints were documented in a patent docket and became part of Correa's internal standard operating procedures. In 1974, the first batch of pharmaceutical-grade PSK rolled off the line, labeled and sealed under good manufacturing practice protocols. The transition from a wild mushroom on a riverbank to a hospital-ready product was complete. For the first time, a compound rooted in folk medicine was now subject to the same industrial scrutiny as any modern drug, ready for clinical testing and regulatory review. Inside the immune system, a silent conversation unfolds every second, a dialogue between the body's sentinels and the outside world. In 2011, Dr. Hailing Liu and her team uncovered a key part of this exchange. They found that PSK, the protein-bound polysaccharide extracted from Trametes versicolor, directly engages a receptor called TLR2 on the surface of immune cells. TLR2, or toll-like receptor 2, acts as an early warning sensor designed to recognize patterns found in bacteria, fungi, and certain natural compounds. When PSK binds to this receptor, it sets off a cascade of signals that ripple through the immune network. The effect is not a blunt force attack. Instead, PSK fine-tunes the body's defenses. Natural killer cells, specialized white blood cells that hunt for abnormal or infected cells, become more active. T lymphocytes, the foot soldiers of adaptive immunity, multiply and sharpen their ability to recognize threats. This boost is especially important in the context of cancer, where the immune system often struggles to detect and destroy tumor cells hiding in plain sight. Dr. Liu's research showed that PSK's interaction with TLR2 does more than simply rev up the immune engine. It helps coordinate the release of signaling molecules like interferon gamma and interleukin-12, which steer the immune response toward targeting cancer cells while leaving healthy tissue untouched. This mechanism helps explain why, in clinical studies, PSK given alongside chemotherapy is associated with improved survival rates, not by replacing standard treatment, but by helping the body's own defenses recover and respond. The discovery of TLR2's role in PSK's activity bridges the gap between ancient herbal wisdom and modern immunology. It provides a scientific foundation for the survival benefits seen in Japanese trials and underscores why a mushroom long valued as a gentle tonic could become a trusted ally in the fight against cancer. A treatment's true worth reveals itself in the numbers. Over the past 5 decades, researchers have amassed a body of clinical evidence around PSK that is rare for any mushroom-derived compound. The published literature now includes more than 70 primary clinical trials and at least 14 randomized controlled studies that meet modern quality standards. When pooled together in meta-analyses, the data encompass over 8,000 cancer patients, most from Japan, but with contributions from Korea and China as well. These analyses focus on patients with gastric and colorectal cancers who received PSK alongside standard chemotherapy, not as a substitute, but as an added layer of defense. The numbers are clear. Across these trials, the addition of PSK to conventional therapy reduces the risk of death by about 22%, reflected in a pooled hazard ratio of 0.78. This relative reduction translates into a tangible benefit. For every 14 patients treated with PSK, 1 more is alive at 5 years compared to those who received chemotherapy alone. In practical terms, 5-year survival rates for colorectal cancer rise from 72% to 79% when PSK is included. For gastric cancer, the best documented studies show a jump from 60% to 73%. These are not isolated findings but the result of systematic reviews, including a Japanese meta-analysis that drew from more than 2,300 gastric cancer patients across 8 randomized trials. The scale of the evidence pool sets PSK apart from most natural products. While some sources claim over 400 studies, the validated clinical trial count is closer to 70, with 14 high-quality randomized controlled trials forming the backbone of the survival data. The rest include observational cohorts and laboratory investigations. Still, the cumulative effect size holds steady across different analyses and cancer types. For thousands of patients, the numbers tell a story of modest but measurable gains. One life is extended for every handful treated. In the world of adjuvant cancer therapy, such absolute improvements are rare, and they underscore why PSK remains a mainstay in Japanese oncology wards. The story behind PSK and its clinical evidence is not without complications. While the published numbers suggest a real survival benefit, the underlying studies reveal a patchwork of designs that deserve scrutiny. Many of the pivotal Japanese trials were open-label, meaning both doctors and patients knew who received PSK. This approach, common at the time, can introduce unconscious bias, especially when outcomes like symptom relief or quality of life are measured. Placebo controls were rare, in part because the taste and appearance of the mushroom extract made it difficult to disguise. Only a handful of studies attempted a double dummy design, and even then the challenge of perfect blinding remained. Another issue is clustering. Most of the data come from a small group of Japanese hospitals, often with overlapping research teams and patient populations. This geographic concentration raises questions about how well the results apply to cancer patients in other countries, with different genetics, diets, and medical systems. Western trials are scarce, and those that do exist are small, often underpowered to detect survival differences. The vast majority of participants in the meta-analyses, over 70%, were Japanese, with the remainder from Korea and China. Study endpoints also varied. While most trials tracked 5-year survival or disease-free intervals, few collected systematic data on quality of life or patient-reported symptoms. Intention to treat analysis, now a gold standard, was inconsistently applied. Some reports analyzed only those who completed the protocol, which can inflate perceived benefit. Dosing regimens were not always uniform, with some studies using 2 grams per day and others the canonical 3 grams. Follow-up periods ranged widely, and long-term safety beyond 5 years remains largely uncharted territory. Publication bias is another concern. Positive results from Japanese trials were more likely to reach print, while negative or inconclusive studies often remained unpublished or appeared only in local journals. Regulatory reviewers in the West have flagged this selective reporting as a barrier to broader acceptance. These limitations do not erase the survival signal, but they temper the certainty that the benefit of PSK would translate unchanged to other settings. As the story moves from the clinic to the policy arena, these caveats become central to understanding why a mushroom extract could become mainstream in Japan, yet remain sidelined elsewhere. In the United States, the path for PSK stopped at a wall few could scale. Bringing a natural compound like this to market as a prescription drug would require a new drug application, a process that now costs between $1 and $2 billion and can take more than a decade. But for PSK, there was no patent protection, no exclusive rights, because the mushroom extract was considered a product of nature, not a novel invention. Without the promise of a protected market, no pharmaceutical company stepped up to fund the trials or navigate the regulatory maze. The numbers speak for themselves. Kureha's PSK generated $357 million in annual sales in Japan by 1987, accounting for a quarter of the country's cancer drug market. In the US, even a fraction of that market would tempt most investors if they could secure a patent. Instead, the only major American research came through public funding. The National Institutes of Health awarded $5.4 million to Bastyr University and the University of Washington to study PSK as an adjunct in advanced breast and prostate cancer. Dr. Leanna Standish led a clinical trial at Bastyr, but even with promising immune results, the study was small and never moved beyond early-phase research. No commercial sponsor followed. Meanwhile, the Japanese data stood as proof of concept, a low-cost, well-tolerated adjunct that improved survival for thousands. In the US, the economic deadlock was complete. Without the possibility of exclusivity, PSK remained locked out of the FDA approval process, its potential stranded on the far side of a regulatory and commercial divide. On a cold morning in December 2025, The UK Food Standards Agency issued a notice that landed with little fanfare but sweeping consequence. Under the novel food rules, all extracts of Trametes versicolor, the same turkey tail mushroom at the center of Japan's cancer adjunct therapy, were to be withdrawn from retail shelves. The agency's rationale was not a safety concern, nor a single report of harm. Instead, the agency cited an absence of documented use in the UK or EU before 1997, the technical cutoff for what counts as a traditional food. This bureaucratic line erased centuries of safe consumption in Asia and decades of prescribed use in Japanese hospitals, reducing the mushroom to an untested novelty in the eyes of British regulators. For Tom Baxter, founder of Bristol Fungarium, the decision was more than a technicality. He filed a formal legal challenge, arguing that the agency's ruling ignored both global history and the lived experience of thousands who had safely consumed turkey tail. The legal brief, submitted in January 2026, pointed out that not a single adverse event had been linked to the mushroom in the UK or the broader European record. Instead, the ban rested on paperwork, not public health. This episode exposes a modern paradox, a compound known for its safety and clinical value. In one country can be rendered inaccessible by the stroke of a pen in another. For those seeking practical answers, the agency's decision is not just a footnote, it is a barrier, one that still shapes who can access a therapy with decades of evidence behind it. Anyone considering turkey tail as part of a health routine faces a maze of options, but a few clear signposts can help. First, a medical disclaimer. No mushroom, including PSK, should ever replace standard cancer care. In Japan's clinical trials, PSK was always given alongside chemotherapy, not instead of it. For those seeking to mirror the Japanese approach, the dose used in most studies was 3 grams per day of pharmaceutical-grade PSK, marketed as Creston. This is a prescription product in Japan, not available over the counter in the US or Europe. Supplement shelves, however, are crowded with turkey tail extracts. Quality varies widely. To get close to the Japanese standard, look for products that specify beta-glucan content above 30%, use hot water extraction, and list only fruiting body, not mycelium on grain. Brands like Real Mushrooms and FreshCap meet these criteria. A typical 60-day supply ranges from $15 to $30. Anything much cheaper likely contains filler or unverified material. Always check for third-party lab testing. Reputable brands post their certificates online. For those drawn to self-sufficiency, home cultivation is possible. Plug spawn, small dowels inoculated with Trametes versicolor mycelium, can be inserted into freshly cut hardwood logs, then sealed and left in a shady spot. Colonization takes 6 to 12 months, and fruiting can continue for several years. While the process is slow, it offers a direct connection to the mushroom's natural cycle. Regardless of the route, remember, supplements are not substitutes for medical care. Each step, from shopping to home growing, should be weighed with both curiosity and caution. Today, PSK remains a standard adjuvant therapy in Japanese hospitals, prescribed alongside chemotherapy, covered by insurance, and supported by decades of clinical data. Yet its absence from FDA approval and the 2025 UK novel food ban reflects a regulatory world where patentability often outweighs practical benefit. The truth is simple. A mushroom once found on a fallen log can still challenge the boundaries of medicine. What barriers truly serve the patient?